right, so thank you for everyone for joining us in our webinar series focused on cross-species cell types. Let's go next slide. So as just a reminder, this webinar is being recorded. In fact, all the webinars in this series has been recorded. So if you've missed any of the previous 4 webinars we've had, please go check them up on our YouTube channel. If you have questions either via Zoom or YouTube, please post them in the chat. You can post them even when you come up with it, even if it's middle of the talk, and I'll save that later for the Q&A portion. If you're watching a recording of this, please go to our community forum, which is in the YouTube description, and you can post your chats there. Next slide. And so today's agenda is Rachel is going to give a quick overview of the cross-species work you can find on the Cell Types Knowledge Explorer, so you can look at the data itself. And then we'll switch it over to Morgan to talk about the cross-species work done here at the Al Institute, why it's important. And then, of course, we want to leave time for you to ask some questions. I know Morgan's really excited here with what people have about cross-species work. And with that, I'll kick it off to Rachel.
Hi, everyone. I'm Rachel Hostetler. I've been in the previous webinars, so if you watch those, welcome back. It's nice to meet you if this is your first time joining us. I'm briefly going to be going over the Cell Type Knowledge Explorer, which is a tool that was created here at the Allen Institute with their collaborators. And I'll specifically be focusing on how you can use the Cell Type Knowledge Explorer to look at cross-species comparisons. And so this is all based on data from a project from the BICCN, which is the Brain Initiative Cell Census Network, where they created an atlas from the primary motor cortex. And in this atlas, we have data from 3 different species. We have data from human, we have data from marmoset, and we also have data from mouse. And this was a very large undertaking. This project ended up resulting in 17 different studies and involved over 250 researchers. And one of the main flagship studies here was featured on the cover of Nature, and this was all done in 2021. And so the purpose of this project was to create a comprehensive cell atlas of the primary motor cortex in these 3 species. And so a number of different techniques were used, different sequencing techniques, different spatial transcriptomic techniques, patch-seq, looking at single-cell morphologies, using transgenic lines for the mice, using different types of AAV labeling. And you might be wondering, why primary motor cortex? Well, the primary motor cortex is highly conserved across different species, which is not necessarily the same for other areas, right? Somatosensory cortex in mouse is very different from human. Visual cortex in human is very different from mouse. And Morgan's going to get into some more of those differences later in the webinar. And so again, this was focused on the primary motor cortex. Over 3 million cells were profiled using all these different modalities across our 3 species. And to sort through all these data, a tool was made called the Cell Type Knowledge Explorer. And what was also interesting between these 3 species is that not only were these 3 species studied individually, the data was also aggregated and studied together. And so what do I mean by that? So 3 different taxonomies were created using this from single cell and single nucleus data in the mouse. We have single nucleus data in the marmoset and single nucleus data from the human. And so 3 separate taxonomies were created from all of that data. And then the single nucleus data was reintegrated and analyzed to create one comprehensive taxonomy, which then compares these 3 different species together. And here we can really see where we differ between our species and where we are more similar. So on the very far left, if you look at the first LAMP5 cell here, we have a 1-to-1 ratio between our 3 different species for cell types identified. But then if you look at some of the other cells in the middle here, sometimes maybe you have 5 cells in human that equal, 2 cells in mice and maybe 1 cell in marmoset. So here we can really see where we're different and which cell types are really conserved across the different species. And so again, to look at all of these data and to interact with it, you can go online to the Cell Type Knowledge Explorer. And you can access the Cell Type Knowledge Explorer by either going to knowledge.brain-map.org/celltypes, or you can go to brain-map.org, click on Atlases and Data, and then click on the Cell Type Knowledge Explorer. And now I'm going to give a brief introduction and how you can access it. So here is the Cell Type Knowledge Explorer. Here's our taxonomy. We have plots from our 3 different species here. Here I'm clicking on the mouse plot. This is a hierarchical plot showing our different levels of taxonomy. So we have our class in the middle, our subclass on the next ring, and our outer class are our cell types. And if you click on the class level here, now we can see information on how many subclasses are located within the GABA-ergic class. We can look at the gene markers that were used to identify this class. We can look at our subclasses within. So now we're within a subclass. So we're in the SST class. Here's a written summary explaining the SST cells. Again, we can look at the different genes that were used to identify these cells. You can also download all of this data. And this was all published online in those 17 studies that I mentioned. We have reference links that direct out to the actual papers. Electrophysiology, morphological data, some spatial data, some epigenetic data. All of that is here at the subclass level. So here we're looking at the SST subclass. And now we're going to jump down to one of the cell types. And these cell types were determined by the transcriptomic data. So here we're in a specific cell type, the SST-HPSE. Here were the gene markers used to determine that. Here's our morphological data for that specific cell type and the electrophysiological data. And what's really special here down at the cell type level is where we see our homologies. So this is what I want to focus on, our cross species. So here is a heat map that's comparing cell types that were also identified in the human and also in the marmoset. We have our similarity scores here. So darker red means the cell types are more similar. And if we look on the human here, if we look and on the marmoset, we can actually see which cells are very similar to our mouse cell type that I clicked on. So here, 0.94, that's very similar, high in similarity score. That is a homologous cell type found in the human motor cortex. Now we're on the human page. You can just click on the heat map. Here's the name of that cell in the human. Here's some information on how that cell is described and which genes were used in the human data. And again, this is homologous to our mouse SST-HBSE cell. And now if we go back to the heat map again, here's some information on how this heat map was generated. And this is also all available through our data links where you can link out to the project summary or get the DOI to the paper that explains this. Again, now we have our mouse on the right-hand side and our marmoset on the left-hand side. If we go and look for our equivalent, our comparable cell in the marmoset, we see one here with 0.94 similarity. Here's the name of it for the marmoset and some information about how the cell was described in the transcriptomic data. And so this is a really nice tool that allows you to quickly just jump between these 3 different species. And again, this is based on single nucleus data, and this features data from the primary motor cortex. And so that's just a brief overview of how you can access this data using the Cell Type Knowledge Explorer. And this is not the only cross-species work that is done at the Allen Institute. This is an example from one specific project, but there are many other projects going on other species. And so now I'm actually going to hand it off to Morgan, who's going to go more into detail and talk more about the nuances of comparing cross-species data.
Great, thank you so much. Share my screen here. Yeah, so that was a wonderful overview of what it looks like once we have all this data processed and visualized and ready to share. I'm actually going to give you a peek under the hood at some things that are currently in progress and not yet on the Cell Type Knowledge Explorer, but hopefully in the future. In general, I'm going to talk about just how we think about comparing across, broad evolutionary distances, individual cell types, transcriptomic signatures. And I'm going to focus on mammals, but I'll say for the Q&A, I've worked on everything from fish to songbirds to parrots. So I'm very happy to take any questions about your species of interest and what kinds of considerations you might want to have for, say, salmon, which has, 6x whole genome duplication. Anyway, without further ado, so here at the Allen Institute, we obviously produce a lot of different data types. RNA-seq, ATAC-seq, PAC-seq, and MERFISH, which you've heard about in some of the previous webinars, which I'd encourage you to check out if you haven't already. So our team, Evolutionary Genomics, now adds an evolutionary dimension to that. What additionally can we learn by not just comparing all these different data modalities, but additionally considering how they've changed across evolutionary time? And Rachel already told you some of the great work that we've been involved in to put together primate and rodent taxonomies. And what I'm going to tell you about now is how we're expanding out across 180 million years of mammalian evolution. And so if we look at the mammalian brain over time, there have been a lot of changes. So just in not just in motor cortex, but also thinking about bats, which have these incredible echolocation abilities, the abilities to see through sound. And that's reflected in a huge expansion of their auditory cortex. Primates were incredibly visual creatures. You know, we have trichromatic vision, and that's represented through a huge expansion in our visual cortex. Likewise, different mammals that have different kinds of motor and sensory abilities, like platypuses with their sensory apparatus, raccoons with their incredible dexterity to get into, you know, your trash can at night, and of course humans with our ability to speak like I'm doing right now, which is not shared by all mammals, of course. All of that is reflected in expansions of different subregions of the motor cortex. Because, you know, all of these cortical regions, like the cell types, can be further broken down into different subregions, reflecting these different specializations that motor cortex, auditory cortex, visual cortex have adapted to allow these animals to perceive their environments in different ways. So the goal of this project is to cover, again, 180 million years of mammalian evolutionary diversity, all the way from the gray short-tailed opossum and armadillo to a large number of Laurasiatherians, rodents, and primates. There is a special focus on primate evolution so that we can start to understand the origins of what makes us special and unique as humans. But species are chosen based on a wide set of criteria. So these are the species we're looking at. We have RNA-seq, single-cell RNA-seq for all of these species, and ATAC-seq, single-nucleus ATAC-seq for a subset of them. And I will say, additionally, for some species of special interest, we're also profiling different subregions of the motor cortex. And the data processing overview, which I imagine you've probably heard about in some previous webinars, but just to go over simply, is, you know, first of all, we have a standardized freezing and dissection protocol. This is actually much more important than you would think, because a lot of the corrections you might have to do later on when trying to integrate across species will also be confounded by, you know, how many different labs, how many different kinds of protocols, and, you know, conditions of the actual tissue itself you're going to have to correct for as well. So the more you can do to standardize up front with whatever data sets you want to compare to, you know, whether it's Allen data sets or others, I'd highly recommend, you know, right at the start, getting that absolutely right. After fact sorting, you know, all of these species, and this is important. Again, I'm just going to try and point out some different cross-species considerations at each of these steps. This is really critical because different species actually do vary in the neuron to glia ratios, and even the excitatory to inhibitory neuron ratios across species and even across brain regions. And in part, this may be just a scaling issue of the brain. As the brain gets larger, that ratio needs to change. But regardless, if you want to have taxonomies that are easier to integrate, we'd highly recommend doing some kind of, you know, new N enrichment. We then do, the sequencing, which you've heard about, clustering, QC to get everything playing nice together. And we're currently using an ML approach to mapping. So mapping on taxonomies from other sources, taxonomies across species, using SCVI and ScanVI, which was co-developed by Nelson Johansson in our lab. And then finally getting to the fun stuff, all the kinds of downstream analyses we can do to start to biologically interpret these incredible data sets that we have the privilege of accessing. So just to give you an idea of, how things, actually look in the raw here, before you do that pre-processing, just all the different variables between individuals are going to be there. And so we do need to do that ScanVI correction in order to get everything playing nice. And at this point, we do start to see some odd things. So, for instance, with these two clusters, you can see they're not really integrating with anything else. So that's going to be important later. We then do different ways of visualizing port quality cells that we want to remove. Exclude to is just one feature we can look at that's looking at gene counts, the doublet scores, things like that, to try to figure out which cells might be worth throwing away. And so that gets rid of this splatter cluster in the middle, and also this spurious cluster over here that was only mapping to one donor, but we still have this funny thing coming mainly from one donor over here. So to visualize what's going on here, we actually found out that this seems to be a dissection error, for instance, where this is probably medium spiny neurons from the striatum. So this is just an example of, the kind of thinking and detective work you can do as you're trying to clean up these data sets. But again, integrating across individuals is of course necessary before you start integrating across species. And this is just showing some ongoing work in this process where we're trying to get some different samples from different labs put together. And our next steps, once we have this full integrated taxonomy ready to go, are to answer basically 3 different kinds of biological questions. So on the one hand, conservation. So like Rachel was talking about, what do all these species have in common across cell types? What are the conserved cell types, but also what are the conserved transcriptional and regulatory programs? Are the same markers that we know within primates or in rodents, how deeply are those conserved as markers of conserved cell types? We're also, of course, interested in divergence. What makes primates special? What makes humans exceptional amongst primates? What makes all of these species individually what they are? And part of that is looking at novel cell types. So there's some interesting cases we published on. For instance, TAC3 neurons have been discovered in primates to be potentially novel and interesting. And additionally, it's important to put primate and rodent divergences in context, which I'm going to talk about in a second. And finally, which the topic I'm most excited about is convergence. So thinking about traits that have evolved multiple times across the mammalian lineage, what kinds of correlates in terms of transcriptomics, epigenomics can we find that correlate with the emergence of evolutionary traits? And so we are hoping to potentially add to this set of species that we're sequencing. And of course, we're very excited to collaborate with other folks that might have additional species they might want to contribute to the project. So these are just some great references I'd recommend looking into in terms of thinking about mammalian brain evolution and primate evolution in particular. But this is important to think about because, you know, when you're looking at any individual difference in whether, you know, really broadly, whether we're talking about a cell type's transcriptomic signature, you know, changes in individual genes, it's important to put things in context. If you're just comparing two species against each other, for instance, human and rodent, you know, or two brain regions, you can't actually tell is something being gained in one species or lost in another. Is that loss unique or gain unique, or is it something that's more commonly gained and lost across species? You really have to put it in a broader evolutionary context to understand how exceptional is that difference you found within species. So here's just one illustrated example. So in M1, there's this finding that you can even read about in textbooks where humans and macaques have been found to have this 2-to-1 excitatory to inhibitory neuron ratio, whereas rodents have a 5-to-1 such ratio. And so this has been touted as a novel primate specialization. When you put things in their evolutionary context, however, that seems to not be the case. And in fact, the rodents are the species that are exceptional, whereas primates seem to be more in line with the consensus mammalian standard. We're looking into this now, and it could be that this is also just an effect of brain scaling to body size. So again, this just highlights that, you know, something that might appear to be a really interesting finding if you're only comparing not even just two species, but two groups, right? Because these are 4 species, but only representing 2 branches. Really, you need to put things in a deeper evolutionary context to understand what is novel, what does make primates unique, what really is the basis of human behavior and genomic specializations. So getting into the third topic, convergence, a topic that I'm very interested in, we're starting to look now at the evolution of dexterity. So primates, of course, have this incredible precision grip, the ability to use tools, and that's evolved multiple times in primates, with cebus monkeys having exceptional dexterity within the New World primates, and then Old World primates like ourselves also having incredible dexterity. And this has been previously connected to a novel pathway within the motor cortex called by Peter Strick and colleagues as the new M1, which is making more of these direct corticospinal projections onto the hindbrain motor neurons for, in this case, the representation of hand. And so the density of that projection in cebus monkeys, which are the New World primate tool users, is much denser, for instance, than in squirrel monkeys, which don't have that same kind of dexterous control. And I've even found a study showing that raccoon also seems to have a denser such projection than, you know, similarly related but not so dexterous puppies. So this could potentially be an interesting area of work to go down. And, you know, a lot of my work has focused on the evolution of vocal learning, which we now have the opportunity to, you know, study in great depth here. And yeah, and basically it's just, you know, I think we're in a really exciting time where we finally have the genomic resources like never before from many species, many cell types, many brain regions, where we can start to understand, this broader evolutionary context for the features we're seeing. And I hope that I've gotten some of you maybe exciting about thinking of some of these questions if you weren't already. And with that, I'd be happy to take questions, which I believe will be delivered from our moderator here.
Yeah, and so if anyone has questions, feel free to drop it up in the chat. I'm also just going to launch a quick survey. If you guys have any opinions or thoughts, it really helps us with this. But yeah, in the meantime, if anyone has questions, both on YouTube or Zoom, feel free to post them in. Oh, awesome. All right, so we got a question. Amazing talks. I'm interested in comparing cell types and specific brain regions between mammals and birds to identify homologous and novel cell types. Is there merit in that approach, or does not involve integration, but directly compare gene expression profiles across species?
Great question. So I'm also very interested in this. And I'll say that, as you probably know from that question, but what I'll share for others who might not know this, comparing birds and mammals presents a special but exciting challenge. Because the organization of avian mammalian brains is very different. So in mammals, we have this 6-layered cortex, right? Where, it's all that foldy stuff, on the outside of our brains. And these layers interact in a certain way. So birds do seem to have the homologous cell types that make up those layers. But instead of being in this layered organization, they're separated into different nuclei, into different centers of the brain. So you have archipallium in the back is like layer 5, midipallium is maybe more like layer 2, 3. I will say there is still some debate about this, but this is my understanding. But regardless, if you wanted to compare mammalian and avian brains in a satisfying way, it would be important to maybe sample and compare across some of these different regions. So to compare layer 5, you'd want to have RA or some archipallial region, and then HVC for your, layer 2, 3, or, and so, yeah, I would say, be curious, be open to exploring some of the different possible homologies, compare different kinds of brain regions and try to understand where do I see the most conservation of marker genes across these cell types, across these brain regions. But, I'd certainly be very excited to see more of that work. Now that we have these data sets, we have the opportunity to start answering these questions in a satisfying way, whereas, in the past, a lot of the argumentation about avian and mammalian brain homologies have not had this kind of amazing data access. So yeah, I would definitely encourage you to explore, and feel free to reach out too if you'd like any assistance.
Awesome. Leave up for chats, questions if any more. If not, I'm sure we're all happy to get a few minutes back of our day.
Looks like there's a follow-up question. So should we compare on the level of subclass or of cell type in the case of substantially different organisms? I would actually start even higher up. I would start at just start with, neurons and glia, excitatory, inhibitory, the major inhibitory classes, the major, layered yeah, just start as high up as possible, and that will, I think help us to understand where do things start diverging. Because yeah, even starting at the level of subclass might be too fine when we're looking across these divergences getting into the 300 million and more.
That's a great question.
Mhm.
Awesome.
Welcome.
Oh, if anyone else has questions that came up later, feel free to put them in the community chat, community forum. Otherwise, happy for people to get their day back. Thank you so much for joining us, and thank you, Morgan, for this great presentation.
Thank you for having me.